Our work is sort of focusing on the bone tissue regeneration and engineering. To be able to incorporate blood vessels in this engineering tissue is very important. Without the blood vessel, those engineering tissue quickly die. In order to improve the survival of the engineered tissue, you need to incorporate the viable and functional blood vessel into the engineered tissues. Through our research, we find that periosteum plays a very important role in repair and regeneration. Periosteum is very well vascularized tissue. It is a membrane that covers the outer surface of the bone. It contains progenitor cells that contribute to bone formation. In our first CTSI project, we propose that we're going to engineer artificial membrane periosteum to help the healing of the bone. We take the fat tissue from the mouse, and then we were able to isolate the progenitor cells from the fat tissue and then make abundant blood vessels in vitro. And these vessels can also incorporate it into the scaffold. We further can identify a specific population of the cells that really contribute to blood vessel formation. So what we're hoping is to do the same thing in the human tissue to isolate the human progenitor cells from the fat tissue and then use it in our tissue engineer process. The challenge that we have is, to translate the animal study to human. In the mouse tissues, mouse is, small and the periosteum is very thin. But the human tissue, periosteum, is much thicker and is much more vascularized. So to translate the mouse data into human, we wanted to be able to generate a vascularized periosteum mimetic. Once you have a better vascularized tissue, it can support those progenitor cell and they will survive better and contribute to more bone formation. This comes the second CTSI project, which allow us to try to identify effective endothelium progenitor cells, which could contribute to the vessel formation. We are trying to isolate endothelium progenitor cells from the fat tissues that was taken from the patient, and those fat tissue normally are discarded, and we collect those tissues and then make endothelium progenitor cells from those tissue and incorporate those endothelium progenitor cells in our tissue construct and then vascularize those tissue congener construct and then make them more effective in the bone formation process. This is some of the preliminary data that we obtained from the patient samples, and we can see there are a large amount of viable blood vessel formation. So this is sort of a piece of evidence to show that our current approach is working.